Hi, good evening, everybody. My name is Nikhil, and I'm a junior from Acton Boxborough. And with my mentor, Dr. Saheli Chaudhary, I'd like to present my project about the scientific communication, how to write a scientific review paper and critically analyze research paper. This presentation will include an outline of my review paper about CRISPR and a critical analysis of my mentor's research paper. Okay, so what is CRISPR? The basic overview of what CRISPR is that CRISPR, or clustered regularly interspaced short palindromic repeats, is a gene therapy technology that research scientists use to selectively modify the DNA of living organisms. Here's the timeline of CRISPR. CRISPR was first discovered in 1987 as an immune system in bacteria. After the discovery of the bacterial mechanism, researchers were determined to figure out what it was. The next couple of years were spent by adapting CRISPR-Cas9 and characterizing it fully. This led to the idea of using it as a gene therapy tool. A major use of CRISPR-Cas9 was when it was used as a COVID-19 detection enzyme, which was dubbed Sherlock. CRISPR eventually won the 2020 Nobel Prize and was awarded to Jennifer Doudna and Emmanuel Charpentier, and is now widely used in the medicinal world, with major breakthroughs such as gene therapy for sickle cell and personalized gene therapy for a child with a rare metabolic disease. Um, so, as I mentioned, CRISPR was found in bacteria, and here's an image that is more into the mechanism of CRISPR in bacteria. First, the virus inserts its viral DNA into the bacteria. Then, a CRISPR-Cas9 enzyme is sent to cut a piece of that viral RNA and implement it into the CRISPR locus. The cut up piece here is a spacer. When the virus attacks again, the locus creates a guide RNA using the spacer. The guide RNA then implements in the CRISPR-Cas9 system. The system then looks for this invading viral RNA and cuts it all up, which halts the infection process. Okay, so here is an overview of the mechanism in CRISPR and biotechnology. So CRISPR is made up of the Cas enzyme and single guide RNA components. The single guide RNA guides CRISPR using the target-specific cRNA. The Cas9 enzyme cuts the DNA at the PAM site and then creates a double-stranded break. The cell starts to repair the double-stranded breaks using one of the two available repair pathways. Non-homologous end joining is the most common way of fixing it because it doesn't use a template. However, this means that it's prone to errors, such as indels. Scientists mostly use HDR because it uses the template to repair the DNA. Thus, it is easier for making the cells have the desired base pair fix in the DNA. All right. So here's some of the major applications in CRISPR. This includes drug development, gene therapy, animal models, biofuel production, and plant biotechnology. All right. So I mentioned two major breakthroughs, and one of the major breakthroughs is a personalized gene therapy for a girl with a rare metabolic disease known as severe carbamoyl phosphate synthase 1, CPS1 deficiency. CRISPR was used in this case to fix a point mutation in the girl's genome. The other major breakthrough was an FDA-approved sickle cell gene therapy using CRISPR. This targets the point mutation in the beta globin gene and fixes it. There are two sickle cell therapies, Casgevy and Lyfgenia. All right. Obviously, CRISPR would definitely have some ethics and concerns. And one of these are the four most common concerns I found, such as there's a concern that designer babies can increase social inequality. There's a concern about access and equity because CRISPR may increase the disparity between the privileged and underprivileged if it's only accessible to wealthy people or countries. There are also unknown consequences because CRISPR can cause mutation when it edits genes. Some of these mutations can be life-changing and might go on unnoticed and get transferred to the next generation and so on. And lastly, there's a concern about autonomy and consent because the future person cannot give their consent in situations like embryo editing. That raises concerns regarding bodily autonomy and individual rights. All right, so I did a critical analysis of my mentor's research paper, and this is the specific research paper that I did. Yeah, so to explain the context of this paper in a nutshell, this paper talks about the novel aspect of a certain deubiquitinating enzyme, JOS1, in hepatoproteotoxicity. When there's proteotoxic stress in the liver, JOS1 gets monoubiquitinated and then gets transported to the membrane. It then deubiquitinates to SOX1, which helps preventing hepatic apoptosis. When the proteasomal activity is blocked by inhibitors, this protective activity of JOS1 is hampered. To understand this paper further, I had to learn about certain methods that are used in regular research work, such as Western blot, immunoprecipitation, site-directed mutagenesis, co-immunoprecipitation, immunofluorescence, siRNA screening, and live dead assay. And lastly, here is my critical analysis of my mentor's research paper. Though this paper had comprehensively addressed all the points of the proposed hypothesis, there are a few limitations and improvements to the study. The first being that the relation between JOS1 and SOX1 has been shown in cells, but not in an in vitro assay. Another limitation is that in this paper is that they talk about the monoubiquitination and polyubiquitination and how JOS1 gets ubiquitinated, but doesn't show which residue it is ubiquitinated and which ubiquitin ligase is responsible. Similarly, in SOX1. The paper also shows how JOS1 is localized in at the membrane using immunofluorescent microscopy, but can also be shown via subcellular fractionation. And for the fifth improvement, you can also show that they interact by using immunofluorescence microscopy. Lastly, the authors have shown the dependence of JOS1 and SOX1 in vitro, but not in animal models, also known as an in vivo model.
Thank you, Nikhil. Thank you so much.
Yeah, so I would like to say something like me and Nikhil, we are working right now on the new paper. He has written quite a bit, and I'm quite amazed as to how quickly he has grasped all the concepts. Even when critically analyzing my papers, he was very quick in learning about all the methods. And we are still working, and we will be working on writing a full-fledged review paper. He has written uh, many, like a few portions of it, and we will be continue working on the review
Yeah.
paper.
That's amazing. I
Yeah.
see that Silda has a question. Go ahead, Silda, and ask.
Uh,
Go ahead, Silda.
Hi, this is Zelda, and this is a question to the
yeah.
student. So if CRISPR has a potential to cure genetic diseases like sickle cell, you mentioned, where should we draw, where do you think we should draw the like ethical line between treating illnesses and enhancing like physical traits?
So I feel like so could you repeat that question a little bit?
So my question is like
Yeah.
where do you think we should draw the line as like an ethical concern between curing actual illnesses, genetic diseases, or enhancing traits?
All right, so I feel like if the like an illness is like hurting the person, right? We should use CRISPR to cure the person so that the person can get better health in the future and it won't like hinder their performance. But however, if you're using CRISPR to like change a gene that like enhance the person's like normal ability, like for example, if you are like a normal person and you like hinder a gene or like change a gene that enhances them to do like extraordinary things, like not fixing an illness, I feel like that's where you draw the line because that's not really like fixing a gene. You're like adding improvements to like the person being using CRISPR.
So you think it should be used as a limitation rather than like just everybody gets to use it? It should be.
Yeah, well, I feel like CRISPR is not like obviously not very available because it's very expensive and not everybody can have it. So that raises like the current concern of like access and equity, right? So I feel like it should be only used for illnesses and not for like high corporations or like very rich people to like enhance their bodies.
Yeah.
I,
So, if I may add, I think the question itself had the answer, like where should we draw the line? Should
Yeah,
we draw the line in enhancing capabilities or in, like curing disease? So enhancing one's ability is a luxury. I believe that. So it's a luxury, and we have a long way to go before we can afford such a luxury in our lives. So for now, it's more of a use in gene therapy. So I think the question
yeah.
itself had the answer. Yeah.
Thank you. Thank you so much. Does
All right.
anybody else have any question for Nikhil or Sahili? I guess no. All right. Thank you, Nikhil.
Yep,
So
no
next
problem. Thank you.